Hello everyone, welcome back once again to this educational channel on biology. Today we'll be discussing the Pahang Bio Paper 2 2021 and this is set 4. So there are two very good essay questions here. Okay, so we'll be using it to revise your Form 5 topics which are Chapter 10 Inheritance and Chapter 11 Variation. Right? So this is a very good way for you to practice and apply your biological knowledge and concepts which you have studied and memorized. Right? And apart from that, you also learn the techniques of answering the essay questions, and also you can write down the model answers into your paper. Right? The Pahang Bio Paper 2, you can access it from websites like SPM Trial Papers. All right. In this video, we'll be focusing on two chapters in Form 5. Chapter 11, Inheritance. The topics involved are Haemophilia, Down syndrome and the differences between them, and then also the inheritance of Rhesus factor, where you will be drawing a schematic diagram. Then the other chapter is Form 5, Chapter 12 on variation. We'll be discussing the causes of variation and learning how to write it in an essay answer. Right? Then apart from that, we'll be learning the techniques of answering essay questions. Right, let's look at the first question. Right? The following information shows the cause of a genetic disorder. Frequent and overexposure to ultraviolet radiation could lead to mutation. Down syndrome and hemophilia are the examples of genetic diseases caused by mutation. A1. How is hemophilia inherited? Okay, so from the word hemo, we know that it's something to do with blood. So hemophilia is a disease where the blood clots slowly or does not clot at a wound. So there's excessive bleeding, right? So how is it inherited? Inherited means how is it passed down? Bagaimana hemophilia diwarisi? Okay, so it is inherited through a recessive allele, a gene or recessive allele, which is symbolized by this symbol X with a H there, small h. Okay, so the allele is actually the small h here, but it's located on the sex chromosome X, so we usually write as X small h. Okay, so it's only found on the sex chromosome X and not on the sex chromosome Y, because sex chromosome Y is smaller. It doesn't contain so many genes. Okay, so it doesn't have the gene for, or the allele for hemophilia. Okay, which is X small h here. Right. Then secondly, state the risks faced by a hemophiliac and explain how this affects his quality of life. Okay, so the risk is, of course, that he will bleed non-stop if there's a cut on the wound, if there's a bruise internally also, he can bleed non-stop. So risk faced by hemophiliac. Point 1, he experiences excessive blood loss when injured because his blood cannot clot at the wound, okay, due to lack of clotting factor. Right? So I think this one should be about two points, because you are explaining so much about it, but never mind. So let's say this 1 point. Then P2, the hemophiliac will become weak, lethargic, and may die. Okay, because of the loss of blood, and also due to lack of oxygen for cellular respiration. Okay, so due to lack of oxygen and excessive blood loss. Now there's the second part here. Explain how this affects his quality of life. So if there are more points, you can mention that it will affect his activities. He has to avoid playing contact sports like football and other vigorous games, which are not safe for him as he can be easily injured by other players, or he can fall and he can get injured. Right, this second question is a hot question. Where you have to make a comparison. State 4 differences between hemophilia and Down syndrome based on these criteria: causes, number of chromosomes, characteristics, and inheritance. All right. So for differences, you can draw a table and then you write down all the titles here: the cause, number of chromosomes, characteristics, and inheritance, following the question. Right. And also underline the important words here in the question. 4 differences. State 4 differences: hemophilia and Down syndrome. Okay, so 1 column is for hemophilia and 1 is for Down syndrome. Okay, to compare directly. So the first aspect is the cause. Now, hemophilia is caused by a recessive allele located on the X chromosome, which is a sex chromosome. So it is actually a sex-linked gene, caused by a sex-linked gene. But Down syndrome is not caused by a recessive allele. It is caused by non-disjunction of homologous chromosomes number 21 that causes the Down syndrome child in the end to have an extra chromosome 21. So this non-disjunction means that the homologous chromosomes number 21 fail to separate during meiosis, and both of these homologous chromosomes move to the same pole, right? And in the end, the gamete form will have 2 chromosomes 21, all right, after the mutation, all right? And then when it is fertilized by another gamete or undergoes fertilization, there will be 3 chromosomes 21 in the diploid cell of a Down syndrome child, right? Now, another point you can add is that the recessive allele for hemophilia is caused by gene mutation, actually. Whereas for the Down syndrome child, all right, it is caused by chromosomal mutation because there's an extra chromosome 21 in each diploid cell, right? For hemophilia, what we mean by gene mutation is that the allele is formed because of abnormal changes to the base sequence, okay, in the gene of this, or in the DNA of this allele, right? Changes to the base sequence like adenine, thymine, guanine, or cytosine in the DNA of this allele, in the DNA structure. Right, so what are the other 3 differences based on these aspects? Now, how many chromosomes are there in a person with hemophilia per cell, per diploid cell? Okay, let's look at the answer. So you can try to write the answer first, pause the video, write the answer, and then later we'll look at the answers. So the number of chromosomes for hemophilia is 46 chromosomes in each diploid cell of a patient with hemophilia, just like normal, except that the gene, there are some changes to the gene. The gene undergoes mutation, right? But the number of chromosomes is like for a normal person in each diploid cell. Now, there are 47 chromosomes in each diploid cell for Down syndrome child because of the extra chromosome number 21 in each diploid cell. Characteristics: for hemophilia, the blood cannot clot at a wound because of lack of blood clotting factor, so the person will bleed excessively when there's a wound. But for Down syndrome, the characteristics are that the patient has a flat white face, slanted eyes, protruding tongue, and mental disability. Okay, so inheritance. For hemophilia, this disease can be inherited or passed down when the gene or allele is passed down. But for Down syndrome child, the condition cannot be inherited. Now, let's review what you have studied about inheritance of Rhesus factor or RH factor in class. All right. So this is a very important part of the video here to help you review and recall before you try the question. Now, definition. Rhesus factor or RH factor is another antigen or protein present on the surface of human red blood cells, apart from antigen A and B, and the Rhesus factor is also called antigen D. Okay, so first of all, say this one, a Rhesus factor. Now, it is an antigen. Antigen is a foreign substance that may have entered the body of someone, all right? So, it is an antigen only if the Rhesus factor goes into a person who doesn't have any Rhesus factor. But in the person who has this Rhesus factor naturally, it's not an antigen to him. Okay, it's not a foreign substance to him. Now, it's easier if you recall that Rhesus factor is a protein. It's just a protein, right? That is present on the surface of some human beings' red blood cells. Not all. So here in the picture, you see a red blood cell. Now, some people on their red blood cells, they have this Rhesus factor, a protein, or we call it antigen D, because we have already got antigen A and B for the classification of the blood group ABO, AB, AB, and O. So in the classification system, the ABO classification system, okay, antigen A and antigen B are involved, all right? That is something separate. Now we have one antigen called antigen D. Okay, so Antigen D is actually the rhesus factor. And what is the rhesus factor? It's just a protein found on some people's red blood cells, not all. Okay, naturally found on some people's red blood cells. So if a person is found to have this antigen D or rhesus factor or this protein, then he is said to be rhesus positive. Okay, so some people have the rhesus factor, they are rhesus positive, or short form is Rh positive. Okay, so not everything positive is bad. Like when you look at if you have tested your blood, you will have a card which says rhesus or antigen D, rhesus factor antigen D positive. It doesn't mean it's bad. And it doesn't mean that it's good. It's just that it's different from a person who does not have the rhesus factor. Okay, so some people do not have this rhesus factor. Now their red blood cells will be something like this, okay, without any antigen D or rhesus factor or the particular protein on the surface of their red blood cells. Okay, so they are said to be rhesus negative. Okay, so there's no advantage to being rhesus positive or rhesus negative, right? It's just a trait. And most people are rhesus positive. Okay, and then there are some people who are rhesus negative. All right, so if a person is rhesus negative, he cannot receive a blood transfusion from a person who is rhesus positive. Later, this one is not in our discussion here, right? We are discussing the inheritance. That's why it's important to classify, blood into two types: rhesus negative and rhesus positive. If the blood of a person is rhesus negative, he cannot receive a transfusion of rhesus positive blood from someone, because the antigen, that's when it becomes an antigen. The rhesus factor will become an antigen to the person who receives the rhesus positive blood, if the person is rhesus negative. So to the person who is rhesus negative, this Rh factor or rhesus factor or antigen D is a foreign substance that flows into his body, okay, through the blood transfusion. And then he will produce antibodies called the rhesus antibody or anti-D antibody to act on and destroy this rhesus factor or antigen D. And then it can cause agglutination of the donor's red blood cells where they stick together. That can cause serious effects for the recipient, for the rhesus negative person. Okay? But anyway, apart from that, let's look at the inheritance. Inheritance of rhesus factor from parents to children is based on Mendel's law, on monohybrid inheritance. It's similar to monohybrid inheritance, okay, which we are going to discuss afterwards. So absence or presence of rhesus factor in a person's blood is controlled by a gene for rhesus factor, and there are two different alleles, two different forms of the gene, okay, and we assign the letters Rh positive, okay, for the alleles. So this term is not just an R, okay, it's Rh positive. So allele Rh positive causes formation of rhesus factor or antigen D, and it's a dominant allele, whereas allele Rh negative does not cause the formation of rhesus factor, okay, it is a recessive allele. Take note that the allele is not just written as an R, a big R and a small r. It's written as Rh positive for the dominant allele that causes formation of rhesus factor, and Rh negative means that it does not cause the formation of rhesus factor. Okay, so that's where it's slightly different from the normal type of monohybrid inheritance. We have to use these alleles, assign these letters. These are the standard. This is the standard way of writing the allele that causes formation of rhesus factor, and this one, Rh negative, is the allele that does not cause the formation of rhesus factor. Then the second thing is the allele Rh positive is dominant to the allele for the allele Rh negative. Okay, so how many different genotypes are there? Now in monohybrid inheritance there are 3 different genotypes, and similarly for inheritance of rhesus factor there are 3 different genotypes. The first one is the homozygous dominant genotype, meaning there are 2 dominant alleles. So Rh positive, and the person's phenotype is that he is rhesus positive. That is his observable characteristic. And the genotype is the genetic content or composition inside the cells. So there's another type of person who is heterozygous, meaning he has one dominant allele, Rh positive, and one recessive allele, Rh negative, okay, in each of his diploid cells. Okay, so why two? Why are there two alleles? One is given by the father and one is given by the mother. Okay, because we are diploid organisms, we are formed from formed through the fusion of a sperm and ovum. So we have two sets of chromosomes in each of our cells. Okay, so we have two alleles in this case for rhesus factor. Now the phenotype for this heterozygous individual is rhesus positive because Rh positive, which causes formation of rhesus factor, all right, is the dominant allele, okay, compared to Rh negative allele. Now the third type of individual is the homozygous recessive individual that has two recessive alleles, Rh negative. So this person does not have any Rh positive allele which is dominant, thus the recessive allele is able to express its trait, and so the person is rhesus negative. Okay, so these are the three different genotypes, okay, and these are the two different phenotypes, either rhesus positive or negative. If you're rhesus negative, you already know your genotype. It must be homozygous. You must be homozygous recessive, having two Rh negative alleles. But if you are rhesus positive, you still do not know which genotype you are unless you go for blood tests, genetic tests to find out. Okay, you could be homozygous dominant or heterozygous. All right. So this picture here shows you the rhesus positive blood with the rhesus antigen or rhesus factor or antigen D on the surface of the blood cell. This is just a depiction of it. And for rhesus negative blood, there is no antigen D or rhesus factor on the surface of the red blood cells. So this type of person who is rhesus negative can produce the rhesus antibody to act against the rhesus positive blood that has entered the body of the rhesus negative person. Okay, so now we are ready to talk about the inheritance of rhesus factor and draw the schematic diagram. Right, let's look at question 10C. Diagram 10.1 shows the D antigen, which is the rhesus factor, okay, found on the surface of human red blood cells. Okay, so this Y-shaped structure, okay, is the antigen D, right, or rhesus factor. Just remember, it's a type of protein. Now, individuals whose red blood cells contain the rhesus factor are referred to as rhesus positive individuals. Okay, so if the blood has this antigen D or rhesus factor, then this person is considered rhesus positive. Okay, positive meaning having that rhesus factor, which is a dominant trait, while individuals who do not have the rhesus factor are known as rhesus negative, and that is a recessive trait. Okay, for example, humans or individuals with blood that do not have the rhesus factor or antigen D, like this one. So then the person is known as rhesus negative. Now, a husband has heterozygous dominant rhesus positive blood, while his wife has rhesus negative blood. Using a schematic diagram, explain the probability that their children have rhesus positive blood type. Okay, so underline, okay, the technique of answering is first you underline the genotypes or the description for the husband, wife, and children. Husband is heterozygous, and he will, of course, show the dominant trait. That is, he will have rhesus positive blood. His wife has the rhesus negative blood, right? And then we are supposed to find out the probability that the children have a rhesus positive blood. That means following the father. Okay. Now you can pause the video a while and try to write your own answers first. All right. Draw the schematic diagram. Now underline here your task word here is to use a schematic diagram and explain the probability. So explaining means you draw the diagram, genetic diagram, and after that you have to explain in words something about this. Okay, how you get the probability that their children, the probability that their children will have rhesus positive blood type. How you find out? Okay, how you determine that? All right, so pause the video a while and try and answer the question yourself. Draw the schematic diagram. Okay, remember you must have all the titles on the left hand side. For example, parents, phenotype, parents, genotype, right? And then meiosis, gametes, and so forth. Okay, so pause the video. Right, now let's look at the answers. Right, so now let us discuss how to draw the schematic diagram for the inheritance of rhesus factor, right? So first of all, we have to determine the genotypes of the husband, the wife, and then after that, from these genotypes, we draw the schematic diagram. We can find the genotypes of their children and then the phenotypes, right? So parents, father, mother here, or husband, then wife. So the parental phenotype is father is rhesus positive as stated in the question, and mother is rhesus negative, right? Then the parental genotype. Okay, the phenotype is the observable characteristic or the characteristic that can be tested. So the blood can be tested to find out whether the parents are rhesus positive or negative. And then from there we write down the parental genotype, which is the genetic composition in the cell. What are the alleles in the father's diploid cells, okay, in each of them. So since the father is heterozygous, he must have one dominant allele, Rh positive, that causes the formation of the rhesus factor, and one more allele, which is Rh negative, that does not cause the formation of the rhesus factor. Okay, so before you draw this schematic diagram, actually you have to, it's better to write the key first, as stated on this left hand side here, all right, where we say let Rh positive represent the allele that causes formation of rhesus factor, and it is the dominant allele. And let the allele Rh negative, okay, let the allele that does not cause the formation of rhesus factor be written as Rh negative, and it's the recessive allele. Okay, so take note that the alleles are Rh positive and Rh negative, not just R. And then we don't use capital letters and small letters to differentiate the dominant and recessive allele. Now we use the symbol plus and minus here, all right, as a superscript at the top here. So father is heterozygous, so he must have one dominant allele and one recessive allele here. Okay, so Rh positive, Rh negative. Whereas mother is rhesus negative, so there's only one genotype, and the mother must be homozygous recessive, meaning having two identical recessive alleles. So Rh negative is the recessive allele. Rh negative, Rh negative. Okay, next, meiosis occurs. So we write here meiosis, and then the next line are the gametes, right? So to find the gametes, we just separate the two alleles from each other and put one in each gamete, right? So for the gamete cell, we draw a circle. Okay, that's the normal way of writing, of representing it. And then inside we put the allele. Okay, so separate the Rh positive allele from the Rh negative allele. Okay, because in meiosis, homologous chromosomes will separate and move to opposite poles, carrying the alleles with them. So these two alleles will separate, okay, during the anaphase 1 of meiosis. Now for mother, if we separate the Rh negative allele from the other one, which is also Rh negative, we find that all the gametes have the allele Rh negative, right? All the alleles have all the gametes have this allele Rh negative. Okay, so we just write one is enough to represent all of them. Now, then fertilization occurs where the male gamete, the sperma, fuses with the female gamete or ovum. So there are two possibilities. The first one is first male gamete Rh with the Rh positive allele fuses with the female gamete, which has Rh negative. So the genotype of the offspring of the child, that means the F1 generation, is Rh positive, Rh negative. Okay? And then what is the phenotype? So below we can write the phenotype. Since there is a dominant allele Rh positive, this child is Rhesus positive. Okay? Then the other possibility is this gamete with Rh negative allele fuses with this gamete which also has Rh negative allele. So you produce a child with the genotype Rh negative, Rh negative. So this child is homozygous recessive, has two recessive alleles, and he or she is Rhesus negative. Okay, that's the phenotype. Right. So after that, we can find the phenotypic ratio. Okay. Here we can see that there's one Rhesus positive child to one Rhesus negative child. So the ratio is 1:1. All right. But for your question, you're asked to explain the probability that their children have Rhesus positive blood. Probability. So for probability, usually we write it as a fraction, and sometimes it's written as a percentage in biology. So let's find out how to write it. So probability that their children have Rhesus positive blood type equals to 1 out of 2, right? Out of 2, 1 is Rhesus positive. Okay? So you can either write half, or half times 100 equals to 50%. Write down both. Okay? So that's the answer. Now, how's the marking for this part? Right? So it depends on the number of marks, but basically each horizontal statement here, or what you write horizontally, equals to 1 mark. For example, you write down the parental genotype. Since you determine the parental genotype yourself, so you get 1 mark for that. If the whole thing here, the components are all correct. And then meiosis, maybe 1 mark, or meiosis plus fertilization together, if you have written these two, maybe 1 mark. Okay? Gametes, 1 mark, right? If all are correct. So write the gametes. When you write this title gametes, all the gametes, the gamete cells, must be in the same horizontal line, on the same horizontal line. Don't have one gamete here and then the other one down here. Okay? Then it's considered not part of the answer already. So all must be on the same horizontal line. And then, of course, the F1 genotype, which you determine, all right, this whole thing is 1 mark. F1 phenotype, 1 mark. And then 1 mark for the calculation and the answer. Please show the calculation. Don't just give the answer 50%. All right. So basically, this kind of schematic diagram will be about 6 to 8 marks, plus the answer for the probability and plus the calculation. All right, 6 to 8 marks. Let's read the last question here, which is related to chapter 12, variation of the Form 5 syllabus. Variation means differences among organisms of the same species in terms of phenotype and genotype. Variation allows us to identify individuals in a population. Diagram 10.2 below shows a picture of the patterns on the fur of the kittens that look different from their mother. Now describe how this situation can occur. 6 marks. Okay, so this is a good HOTS question. All right, because you have to apply your knowledge to a new situation. So you can see that the pattern of the fur and even the color of the fur is different from the mother. The mother doesn't have much of the patterns here on the body, but the kittens do have. So what is the cause of this variation or differences? Right, it is the genetic factor, the genes, because when the kittens are born, they already have this fur pattern that's different from the mother, okay, which is determined by genes. So the genetic factor is involved to cause variation, right? So think for a while, what are the genetic factors that cause variation? There are 4 genetic factors. Now, are environmental factors involved here? Not exactly, because when the kittens are born, they already have these fur patterns that is determined by their genes, right? So unless they're talking about the size of the kittens and the weight of the kittens compared to each other, that can be caused by environmental factors, if the amount of food they eat is involved. But in this case, only the genetic factor is involved in the determining the fur pattern on the kittens. Okay, so think about the genetic factors and try to write an answer, okay, before we discuss further. Now, what's the first thing you should do before you write down your answer in the answer script? So I've asked many students what they do when they are going to start writing the essay. I say, do you plan your answer by writing out the outline first, or straight away you write in the answer? And many of them tell me, I think about 80% will tell me, Cikgu, hentam sahaja. That means straight away write the answer inside the answer script. Okay? So actually, one of the tips for answering essay questions is that you should plan your answer first by writing out the outline, the rangka jawapan. Okay? Write it on a rough piece of paper, okay, and let your brain brainstorm. Think freely about what the answer can be. Okay, in this case, what are the genetic factors that cause variation? So your brain will start to recall and get all the facts out. Okay, the genetic factors, recall the genetic factors from its memory box, right? And then you can write it inside here. And if you can recall an acronym to help you open that memory box and write out all the keywords, then even better. So in this case, I have an acronym called CRIM, C-R-I-M. C for crossing over, R for random fertilization, I for independent assortment of homologous chromosomes, and M for mutation. So these are the 4 keywords that will get you the marks, 1 mark each, right? So if you can recall this, write it in first in your outline, then you know that you have the basic answer there. After that, you can plan your answer well. Okay? Now, without an outline, sometimes students will write in a confused way, all right, and they may miss out some factors, genetic factors, okay, or their answer is not systematically arranged, all right. So write an outline first, and it may take you about 1 to 3 minutes like that to quickly scribble it on a paper, or even on a rough piece of paper, all right. That is the tip for answering essay questions, okay, writing out an outline first. Right, let's quickly review this subtopic, causes of variation, before we discuss the essay answers. So this topic is found on page 261 in your Form 5 textbook. You can refer to this page, right? 12.1 causes of variation. So variation means the differences in characteristics between organisms of the same species. And there are 2 causes of variation. Generally, there are 2 causes of variation. A, genetic factors, and B, environmental factors. For genetic factors, there are 4, okay? 4 events that lead to, that causes variation. The first one is crossing over. Second one is independent assortment of homologous chromosomes. So these 2 occur during meiosis. Third one is random fertilization, and fourth one is mutation, right? So we can form the acronym CRIM, C-R-I-M, for these 4 factors. C for crossing over, R random fertilization, I independent assortment of chromosomes, and M mutation. Next are the environmental factors. Examples are temperature, sunlight, pH, and nutrients, okay, that may cause variation among organisms of the same species, okay. So this one will not be discussed in this essay question, right? But we can form the word peanuts with it. P for pH, NU for nutrients, T for temperature, and S for sunlight. So think of cream peanuts, or peanut cream, right? To remember the causes of variation, peanut cream. Right, so now let's discuss the genetic factors in more detail, as we're going to use this in our answers afterwards. View of the genetic factors that cause variation. The first one is crossing over during prophase 1 of meiosis, right? So here we see a paternal and maternal chromosome, and paternal chromosome has dominant alleles here. Maternal chromosome has recessive alleles. Now, so during prophase 1, the non-sister chromatids of the homologous chromosomes here break at the chiasma. Chiasma is the point of contact, the point of crossing over. So where they touch here, the chromatids will break, and then segments of chromatids will exchange places. Okay, so non-sister chromatids are the chromatids from the different chromosomes. One from paternal chromosome, one from maternal chromosome. Okay, so the segments of chromatids, okay, will exchange places, okay, when they break at the chiasma. And here we see that now this chromatid here has a segment of the maternal chromatid, right? And for this chromosome here, the maternal chromosome or chromatid, it has a segment of the paternal chromatid. Okay, so the exchange of segments of chromatids, and this leads to new combination of genes or alleles. You can see here, this is a new combination. There are dominant alleles and now also recessive alleles, okay, in this chromatid, and likewise for this one. Okay, so at the end of meiosis, all these chromatids will be found in different gametes when the sister chromatids separate from each other and move to opposite poles, right? So now you have 4 chromatids which have different combinations of alleles, all right, between them. And these 2 are the new combinations of alleles, okay? So basically, crossing over leads to the exchange of genetic material, which results in recombinations or new combinations of genes. This produces genetically different gametes and causes variation. The second genetic factor that causes variation is the independent assortment of homologous chromosomes during metaphase 1 of meiosis. For example, during this metaphase 1 of meiosis, each homologous chromosome pair, such as this one here, which consists of 1 paternal chromosome, the blue one, and 1 maternal chromosome, the red one, is arranged randomly, secara rawak, disusun secara rawak, on the equatorial plane of a cell. Okay, and the way it's arranged is independent of the arrangement of other pairs of homologous chromosomes, like this second pair, which is a shorter pair. Okay, so that's what it means by arranged randomly, and also by that's what it means by independent assortment of homologous chromosomes. So in the end, these 2 paternal chromosomes, paternal chromosome number 1 and paternal chromosome number 2, will move to the same pole and be found in the same gamete. So the first gamete only has paternal chromosomes in it, paternal chromosomes from this individual, and then the maternal chromosomes are facing the same pole. So at the end of meiosis, these 2 maternal chromosomes will be found in the same cell. All right, so the second gamete has only maternal chromosomes. But is there another arrangement of the homologous chromosomes at the metaphase plate? Yes, okay, because of the random arrangement of homologous chromosomes, you get the second arrangement, where the paternal chromosome number 1 faces the same pole as maternal chromosome number 2. And for this other side here, which is different, maternal chromosome number 1 faces the same pole as paternal chromosome number 2. Okay, so if the arrangement of chromosomes is like that, then it will produce 2 genetically different gametes at the end of meiosis. The third combination of paternal and maternal chromosomes is in this gamete, right? It has 1 paternal chromosome number 1 and 1 maternal chromosome number 2. And the fourth combination of paternal and maternal chromosomes is seen in this gamete, which has 1 maternal chromosome number 1 and paternal chromosome number 2. Okay, so altogether you have 4 genetically different gametes produced from this cell with 4 chromosomes, all right, when there is independent assortment of homologous chromosomes, all right, and the genetically different gametes produced will then cause variation to occur, all right, if they are fertilized. So for a cell that has more chromosomes, then there are more possible arrangements of the homologous chromosomes on the equatorial plane of the cell. during meiosis. The third genetic factor that causes variation is random fertilization. So in the female reproductive system, fertilization between a male gamete and a female gamete, okay, between the sperm and the ovum, occurs randomly, also in the cats, in the female cat. So an ovum in the female cat, okay, can be fertilized by any sperm out of the millions that are present. Right? So each zygote produced after the fertilization process is unique and genetically different from other zygotes, and also genetically different from their parents, from the mother cat. Right, so we have already written out the outline for this answer, right? CREAM, the acronym, and then genetic factors which cause variation are crossing over, independent assortment of chromosomes, random fertilization, and mutation. So we are going to plan our answer according to this scheme or outline, all right? Right, the first point, P1, genetic factors cause variation or differences in fur pattern between the kittens and mother, right? So we start with the first factor, crossing over. So for all the factors, please split them up into different paragraphs, the explanation for the different factors. Now, crossing over, the explanation for crossing over should be in one paragraph, and then the next paragraph should be for independent assortment of chromosomes, and then the third paragraph for fertilization between sperm and secondary oocyte. So don't write all into one big chunk without any paragraphs, because it's very difficult for the examiner to identify the keywords sometimes, if they are marking in a, with a deadline, okay, to meet. And that is my feedback to you as an examiner, all right, or as a marker, that you should break up your answers into paragraphs, okay, so that it's easy for the examiner to find the answers, all right, and to give you the marks, okay. So write in paragraphs. Now, crossing over occurs during prophase 1 of meiosis, okay, that's the first point. Then explain the result of this crossing over. So it causes exchange of genetic material between the non-sister chromatids of homologous chromosomes. This results in new combinations of genes in the chromosomes. Okay, and the final result is to produce a genetically different gametes in the parent cats, which are the father and mother cats, right? So always refer back to the question, and relate to it. Now, the second factor, so a new paragraph, is independent assortment of chromosomes that occur during metaphase 1 of meiosis. This leads to new combinations of paternal and maternal chromosomes in the gametes, and again it produces gametes which are genetically different, or with different genetic content. Now, the third one is fertilization between sperm and secondary oocyte or ovum. It occurs randomly in the mother cat. So any sperm can fertilize the ovum, all right, and this will lead to new combinations of genes in the zygotes. Now, all these factors, okay, the three genetic factors, produce zygotes and offspring that have different genetic information from the parent, from the mother cat, and also from the siblings, okay, because of the genetically different gametes that are formed, okay. So any 5 points here to get 5 marks, okay. So there are many points for you to get the 5 marks. Now, one last genetic factor that causes variation is mutation, okay. So P11, gene and chromosomal mutations may occur in the gametes of the mother cat if she is exposed to mutagens such as gamma rays and food preservatives. It can also happen in the father cat. So mutagens are the substances or the rays that cause mutation, okay. And what happens in mutation? So Mutation are sudden abnormal changes to the genetic material in cells such as the gamete and other cells. So these cause changes to the genetic material of a gamete, such as changes to the base sequence in nucleotides of a gene, or just changes to the base sequence in the DNA molecule of the gene, right? In the deoxyribonucleic acid molecule in a gene, all right? So if this happens, it's called gene mutation. And changes in structure or number of chromosomes are chromosomal mutations. Okay, for example, as you can see in this picture, the UV rays, or it could be gamma rays or some chemical substances which are dangerous, which are mutagens, could damage the DNA, the structure of the DNA molecule in the gene, right? So it could cause the bases, one of the bases to be deleted, to drop out, and this is called base deletion, right? Or maybe a base could be inserted, added into the sequence of the bases. Okay, so if these things happen, then the genetic code of the gene is changed. The code to produce the proteins are changed. Okay, and then it will cause the mutation, a change in the phenotype or in the trait of the kitten, in the fur pattern of the kitten, right? So this is a gene mutation, one cause of gene mutation. Now, these mutations produce the kittens with different genetic content and fur patterns compared to the mother cat, okay? Because the mother cat didn't undergo the mutation, but the gametes did. So when the gametes are fertilized, then you produce the kittens which have the mutation, and it could result in different fur patterns compared to the mother cat. So we have reached the end of the video. I hope that you have learned something from it, and please like, share, and subscribe. Now, before I end, the secrets to success as I see it, right? It includes believing in yourself, learning from mistakes, being teachable, to learn the right things, and being accountable. Having a beautiful vision in your mind of your dreams, having the habit of motivating yourself to reach your goals through the way you talk to yourself, all right, and through always referring to that vision, that beautiful vision in your mind, all right, to motivate yourself. Being persistent to try, no matter what obstacles you face, and no matter what failures we may face, all right, or experience. Now, which one of these resonate with you, right? So if you have any other secrets to success, do share in the comments section below. Right, all the best in your studies, and goodbye for now.